Now we'll turn our attention to acellular pathogens. We'll take a look in this video at viruses. If we look at some of the characteristics of viruses, we see that they are referred to as infectious acellular pathogens. Acellular, without cells. Um, Viruses are not included in with any of our phylogenetic trees because they are not considered to be alive. They are not composed of cells. In order for viruses to metabolize or reproduce, they must first take over a host cell. So they are referred to as obligate intracellular parasites. They have to live inside of a cell in order to achieve their ultimate goal of replication. The genome, the genetic material of the virus, will enter the host cell and basically take over the host cell. Instead of the host being able to, produce its own proteins and do its own thing, it's going to start cranking out parts of the virus and produce these new virus particles called virions. Then those are assembled and then released outside of the cell. Viruses have a specificity in terms of what type of host and what type of cell they can infect. When we look at their genetic material, they will have DNA or RNA, never both. If we contrast that with our cellular parasites that we've been talking about, we know that living cells have both DNA and RNA, but viruses do not. Their genome, so their genetic material, is surrounded by a capsid made of protein, and then in some cases there may be an envelope that surrounds the capsid. They have a very small genome in many respects because they don't have to have all of the genes that code for setting up the machinery. Because they take over host cell machinery, the genetic material they do have is very specific to just taking over and cranking out more viruses. Viruses can infect every known type of cell, but most viruses can only infect a very specific type of cell, and this is called its host range. For example, we have a complete group of viruses called bacteriophages or phages, and these viruses can only infect bacteria. They can't infect, our cells. They can't infect plant cells at all. They only can infect bacterial cells. Viruses can be transmitted in a variety of ways, just like other pathogens can. And later on in the semester, I'll do a specific lecture on all of the different modes of transmission. Viruses can cause a wide variety of different types of infections and diseases. They can be transmitted between different types of animals. These are called zoonotic diseases, where we have a virus that can be transmitted from, let's say, a dog to a human. So viruses can jump species. Viruses are too small to be seen with a regular standard light microscope. We have to use electron microscopes typically in order to even see them. So what you're seeing here is an electron micrograph of a particular type of bacteriophage. And so with the bacteriophage, we see the genome here. We see this protein capsid surrounding it. And then bacteriophages have a couple of other structures. They have this sheath plus these little tail fibers that allow them to kind of anchor to the surface of the bacterial cell So bacteriophages kind of look like little spiders or some type of little insect to me. And I can just picture them kind of crawling around the surface of the bacterial cell until they find their specific receptor. So viruses, again, tend to be smaller than bacterial cells, than eukaryotic cells, and typically seen with just the electron um, microscope. So remember that viruses are composed of their genome, their nucleic acid, either RNA or DNA, plus a protein capsid and maybe an envelope. So those that just have the DNA or RNA and the capsid are referred to as naked viruses or non-enveloped viruses. And those that have that lipid layer are referred to as enveloped viruses. The envelope is actually a portion of the phospholipid membrane that is obtained as the virus leaves the host cell. So it takes a piece of the host cell's membrane with it when it leaves. So here we have a naked virus. We have the DNA here in the middle. We have the capsid on the outside. Then we have these little spikes that are sticking out. These are typically made of glycoproteins, which are proteins that have some type of sugar or carbohydrate attached to them. Here is an example of an enveloped virus where we see our genetic material here in the middle. We've got our capsid, and then we have the envelope around it, and then again we have some glycoprotein spikes on the outside. And typically the purpose of these spikes is to allow the virus to attach to or dock with the host cell. Now, the shape and size of the capsids can vary. We can have helical, polyhedral, or complex. The helical capsid forms the shape of the tobacco mosaic virus. So here's a picture of the tobacco mosaic virus. And you can just kind of see that it's this very unassuming kind of long cylindrical or rod-shaped structure, and the genome fits inside the length of the capsid. Polyhedral capsids, like this example here in the middle, that is the human rhinovirus, one of the causative agents of the cold, is many-sided. It's almost geometric, and one of the most well-known or well-studied is the icosahedral capsid. It kind of looks like a soccer ball, where it has these geometric shapes that all link together. Helical and polyhedral viruses can have envelopes or be naked. And then our complex capsids are like the bacteriophages or like pox viruses. They have an appearance that's not helical and not polyhedral. There are two different ways that we can kind of classify and organize viruses. We do need to have a way to classify them, even though they aren't considered to be alive. But we have the ICTV, the International Committee on Taxonomy of Viruses, and they have developed a nomenclature system that classifies viruses into families and then genera based on their genetics, what type of DNA and RNA they have, their chemistry, their shapes. There are 7 orders, 96 families, and 350 genera, and you'll see this as we go through and study the different viruses. Our families end in viridae, so pox viridae, herpes viridae, and then the genus name ends in virus, like herpes virus, for example. An alternative to this is what we refer to as the Baltimore classification system. The Baltimore classification system classifies viruses based upon their genome. Do they have DNA or RNA, and what type? Is it single-stranded or double-stranded? And then their mode of replication. So we have both means of classification, and we will really use both. We'll use the naming conventions set forth by the ICTV, but we'll also take a look at the specific genomes based on the Baltimore classification.